So welcome once again. This is the Getting Started with Greenline webinar. Today, the purpose over the next hour or so is give you an update and an introduction to DNA Subway Greenline. We know that many folks may be familiar, but I know a lot of folks aren't. So I have a couple of slides which will also be available. There is one link that I'm also going to put into the chat right now, which is the CyVerse Learning Center, which is available to you at learning.CyVerse.org. So I'm just going to throw that into the chat. And I will also send a follow-up email, but at learning.cyverse.org, which I'll show a little bit later in the presentation, you have the ability to view not only the video, and I prepared a separate video just on Greenline in 5 minutes, going over the highlights, but there's also a full tutorial that you can follow and use in class in any way that you see fit. So with that said, I am going to go ahead and let's just make sure. Some folks are talking about echoes. Just want to make sure that everybody has microphones off. You shouldn't have the ability unless Zoom changed something, but just make sure your microphone is off if you do have a microphone. That should reduce any echoing. And we have folks here who are monitoring my presentation, so if we do have any other audio or technical issues, they will attend to it and let me know, and we'll correct those as soon as possible. Okay, so let me start by showing you just a couple of slides, give you a little bit of an orientation, and as we do, I'll go back and forth between the slides that give you a general overview and show you some specific features of the new and updated Greenline with DNA Subway. So let me go ahead and share my screen. And I'd like to go ahead and take a look at the Subway slides. Okay, so as you may be familiar with, this project is brought to you by CyVerse, which has the mission and the vision of transforming science through data-driven discovery. Some of you may have only really used some of the capabilities of DNA Subway and not known about the other things that are available to you, CyVerse. Towards the end of the presentation, I'll give you a little bit of a detail of how actually Subway connects very nicely to the CyVerse resources. One important thing we want to advertise and let people know about is that we're funded by NSF. So it's really a collaborative project. It's been renewed back in September. We'll be around with that funding for the next 5 years and beyond. So we want folks to know that the resources are available to them. If you don't know about CyVerse, it's really suitable for all types of data. We're going to be talking today about RNA-Seq. I'll give you a little bit of a preview of the purple line, which is going to be for looking at metabarcoding, metagenomics at the very last end slides that I have. But it's really a bit for data, and I encourage you to take a look at some of the other capabilities that are there. For those who may not know what some of those are, one of the ones that we're using today is the data store. One of the advantages of using DNA Subway Greenline is that you have the ability to look at large amounts of data. So we know a realistically sized RNA-Seq experiment may have several gigabytes of data, and you can take care of that using the underlying CyVerse resources, such as the data store. Of course, we'll be focusing on DNA Subway, but I'll actually point you in the direction of some of the other resources you may want to do as you do even more sophisticated things in your research or in your teaching. Okay, so today's subject is the Greenline, which we have now completely redone in many ways. The two most important ways that we've redone the Greenline is that we've really changed the workflow that we're using. We're using something now called Kallisto and Sleuth. Some of you may be familiar with those software tools and use them in other contexts, and now we've implemented them in Subway. And they have the advantage of both being very fast, so decreasing the amount of time it takes to do an analysis, and also accurate, and I'll point out some of those features in just a moment. The way fundamentally that those software works is by using a transcriptome instead of a regular genome alignment, and also through a process called pseudo-alignment. I'll give you a little bit of an introduction to that, but I won't really go too deep today. The other thing that we find very exciting is that we've implemented the way that you view and visualize the results in an R-Shiny app. So many of you may be familiar or heard of R as a really fantastic language for statistics and for visualization of data. And really that moves us in the direction of thinking about RNA-Seq, and in fact, all of biology as really experiments in data science. So we'll be going in that direction. So what about Kallisto and Sleuth? And again, not only will this be recorded, but I'll post the slides so anybody can take these apart and remix them in their teaching. This is a quote from a paper I really like. I believe this is from 2016. And this quote kind of encapsulates one of the things we need to think about when we're doing an RNA-Seq experiment. The very first thing in the abstract it says is that RNA-Seq has a wide variety of applications, but there's no single analysis pipeline that can be used in all cases. So in the former constitution of the Green Line, we used the Top Hat tuxedo, which I'll mention in a moment. Now we've moved to Kallisto, which really covers a lot of bases, but may not cover every base. Conceptually, since the DNA subway really is a teaching tool, we feel that we've really placed down the fundamental things you'll need to think about in introducing some of the key topics of RNA-Seq, and also some of the topics thinking about things like big data and how that works in biology currently. Previously, as was mentioned, we used something called Top Hat and cufflinks. Now, there are actually new implementations of the Top Hat workflow, which one could use, but there's several reasons why we chose to use Kallisto and Sleuth. Traditionally, just as a reminder, whether you're new to RNA-Seq or you're refamiliarizing yourself with the way that we previously worked, as you can see from the diagram here, we would start with a FASTQ file of some reads. Then we also would need an annotation. We would need to combine those things to create this SAM-BAM file, this alignment file, which can get quite big. Then we use cufflinks to merge those things and end up with an idea of what our transcripts are, our isoforms are, and then do some type of testing. So that required alignment to a reference genome. It's a splice-guided awareness, but it is actually a little bit slower and computationally expensive. There's still many good methods. You wouldn't use the original Top Hat and cufflinks, and in fact, that's why after this summer, we'll no longer be supporting this on the DNA subway, because the newer methods, even the new Top Hat and cufflink methods, are much more accurate. And in fact, the authors of those previous software packages encourage you not to use them anymore. So we want to make sure that what we're presenting to you is not only up-to-date and accurate, but really the best practice. So this new tool, Kallisto and Sleuth, is based upon a concept called pseudo-alignment. I don't want to go terribly deep into this because I could spend an hour explaining it, but it is nice for you to know a little bit about it and be pointed in the direction of some papers you may want to read, because of course, if you're teaching on this, you want to get at least a little bit familiar with some of the concepts so that you can explain it effectively. The key paper for understanding how Kallisto works is this one that's on the screen, Near Optimal Probabilistic RNA-Seq Quantification. And the idea of pseudo-alignment was, I believe, primarily Nicholas Bray, who's the first author here. Let me summarize it in just 2 slides, I think, just to give you an idea of where this is going and also why we made the decision to upgrade the DNA subway using this approach. So this is a figure that's taken from the paper. And just to give you a little bit of background, because this is a complex figure, let me just give you maybe 1 or 2 things you need to know about it. So in this section A, you can imagine that if you have some real isoforms or real RNA-Seq transcripts that exist in the cell, you've done the RNA extraction, you have that RNA, there physically are these transcripts that exist somewhere in that tube once you extract them. When we actually go through the process of sequencing, in most cases, if we're talking about the short read sequencing of Illumina, let's say, which is still quite the standard, you're going to get a read that may cover some, but not all of those transcripts. So that's represented here in black. Here, A, B, and C, this pink, blue, and green transcripts are the real transcripts, and black is actually the read that you might get, any given read. Of course, there's millions of them, but we'll focus on one. One of the key insights of this thing called pseudo-alignment is that actually, when we're doing an RNA-Seq experiment, what we really care about is getting a count. How many times does a particular RNA-Seq or RNA transcript appear in our sample? And the conceptual idea is that if we look at sample A and sample B and count the transcripts and there's a difference, and that difference is significant, well, that may give us some biological insight. Traditionally, when we have done RNA-Seq and we did alignment, well, actually the way that the software developed was much in the same way that software for genome alignment worked. In other words, there was a matching of nucleotide to nucleotide, and all of those, let's say if you had 150 base pairs in a read, you really want to match carefully each one of those 150 base pairs. But actually, in order to do an RNA-Seq experiment and to do those quantifications, you don't need all of that information. You really just want to know whether that read came from a transcript or did not come from the transcript, and make a count thereabout. So what you're actually seeing in this B, C, and D on the diagram is that what we do is we chop up the read into k-mers, short regions of the read, which we just take advantage of, and look and ask the question, could that small piece of the read, not the whole 150 base pairs, but maybe 30 base pairs, could that have been generated? Is it compatible with any given read? And in this diagram C, the first k-mer, let's say this region represented in this black dot, matches all three transcripts. The next one matches all three. But by that third k-mer, there is a match to the first two transcripts, but not a match to the last transcript. Without going in more sophistication or depth than that, I'll encourage you to read the paper. What this ends up doing is that by the time we get to this little diagram, it's a little bit small at the bottom of E, and again, go read the paper, what you end up doing is getting an idea of, for any given read, which are the transcripts which are compatible by it. Because you're not matching every single nucleotide, you can actually give up counting a lot of things that you don't have to count. You don't have to match every single nucleotide and test, did it match, did it not match? This saves a fantastic amount of time and computational power. An analogy I like to use is in this next slide. If you're looking at this slide on your screen and you're a native speaker of English and even non-native, I've seen and done it in classes, you can probably read this pretty effectively. Now, the only key to every word on this page is that the first letter and the last letter of the word is the same. And it turns out in English and most languages, I suspect, where you have an alphabet, as long as you don't change that first letter and last letter, there's enough context that you can actually read pretty fluently, a paragraph of text. And the same thing actually happens on a non-trivial level with k-mer counting. And oftentimes when we're looking at a transcript, just looking at the first k-mer and the last k-mer is enough to determine whether that read came from one transcript or another. Read the paper, listen to this again, think about it, but essentially what that does is reduce the amount of computation. You're not matching nucleotide by nucleotide, but you're throwing away a lot of information that you don't need in order to just get an idea of whether this count can be called for one transcript or another. Now, the result of that, and this diagram also gives you a little bit of an idea, is that you have several-fold decreases. When we're looking at the bottom here with sailfish and Kallisto, several-fold reduction in the amount of time it takes to do this. So in my experience, and what we hope to really achieve, is that while analyzing data on the green line, using the test data set that I'm going to show you, it took me about an hour from end to end. So from FASTQ files all the way to some analysis, which makes it a lot more tractable for the classroom. I'm sure there will be bigger data sets and exceptions to that sort of hour rule, but for those of you who may use the green line in the past, you may be familiar that sometimes it could take days. Most of that time actually spent waiting for HPC. And there's another speed up that I'll talk about, which is relevant to how we got here today. So let me just summarize that with a little bit of background science, just enough to try to get you oriented and comfortable with this. But when it comes to doing a genome alignment, which is the old way, still valid using the new top hat stuff, but versus pseudo-alignment, there's some differences. Pseudo-alignment's fast. There are some reasons why you might not want that method. For example, if you're looking for novel transcripts, you can't identify anything that's not in your transcriptome. If you're looking at SNPs or other types of discovery, those also would be not something that Kallisto in general would be ideal for. There's some other versions of it that help with some of these problems. But no matter what, think through your choice, document what you do, and think about this as a really fantastic and we hope a streamlined introduction to using in the classroom. Okay, so let's talk about what I'm going to attempt to do over the next 45 minutes or so. Look at my watch. We are going to attempt to go through a tutorial, pull out the highlights. I'll be paying attention to questions. I may not answer every one of them in that second, but towards the end, we'll have some time for any questions that may have accumulated. We're looking at identifying some differences in gene expression between human neuroprogenitor stem cells, some of which were infected with Zika and some were given a mock treatment. So we're going to create a project. We're going to show you how quality control of those reads happens, how the pseudo-alignment works, and how to look at the differential expression or differential abundance using Sleuth. The example data and paper are all publicly available, and I'll point you again to the learning.cyverse.org site where we have all of that. One reminder again, before we go into this, although we do want everybody to start using Greenline and giving us feedback about things that we can improve, just a reminder to use the resources wisely. So another speed up that's made this possible is we're using the cloud. So we're making use of a spin-off project, but a separate project from CyVerse called Project Jetstream, which is a large open science cloud. If you're doing education, research, or teaching, I really encourage you to look at that. But just because you can click a button on the green line doesn't mean you should click every button on the green line, is essentially my message. Probably the smartest way is to do an analysis when you need to do that analysis and make it public so you can share that with everybody. We want to minimize redundancies so that you're not analyzing the same data when it's really not novel data, and having 60 students analyze the same thing when they can share a project, that's a lot more computationally efficient. And I'll show you how to make your project public so that you can share it. The other thing is, including today's webinar and also in the near future, we'll have lots of surveys on the DNA Subway site. We want to know more about your experience and also how we can improve it. But there's always a contact us link at the bottom of the Subway page I'll point out, so you can give us feedback at any time. Many of you may have my email address. I know many of you, perhaps personally, but make sure you use the contact link. That way your email or your message will go to multiple people in case I'm not there at the moment. Okay, so now as we get closer to actually using this, what do you need to get started? Well, the very first thing you need is some input data, and there are really two options for that. One is you may have already some FASTQ files that you have generated in the course of your own experiments. Those are perfect. You can use them, either the FASTQ or the compressed FASTQ.gz. The other alternative, and this is a good alternative for many folks, is to go to the NCBI Sequence Read Archive. And on the Sequence Read Archive, you can search by organism, you can search for RNA-Seq experiments, and pardon me, you can actually download FASTQ files, although you'll have to do some processing to extract them from the NCBI SRA format. You can go ahead and import those files and use them in your Subway project. Although I don't have time to show you right now, if there's extra time or you have a question, let me know. I'll see how much I could show you. Pardon me. But you can actually go to learning.cyverse.org, and there's a tutorial there on how to import data from the Sequence Read Archive. Once your data are in the CyVerse data store, you can go ahead and use them in Subway. Okay, so here's the first thing I am going to show you, just a little bit of a preview of, just to orient you so you know how it looks if you haven't done it before. The best way, if you're working with your own files, to get those files into the CyVerse data store and thus ready for DNA Subway, is to use a tool called CyberDuck. So what I'm going to do is take a look here, and let me just go ahead and show you. Well, there's 3 steps. You're going to download CyberDuck and a configuration profile. You're going to configure it, and then you're going to upload those reads. Let me go ahead and show you some of that. Okay. And also, I saw a chat message. I just want to check that quickly, that nobody's having problems. Okay, that's a question I'll come back to in just a moment. Let me show you the Learning Center. So this is it, learning.cyverse.org. If you just type that in, or I put this in the chat earlier, there's a whole bunch of tutorials at learning.cyverse.org. The first thing is, if you are using, we talked about getting your data in, so you go to this one that's called Data Store Guide. And on the Data Store Guide, the first tutorial there is how to drag and drop and transfer data using CyberDuck. So I will not go through the process of downloading CyberDuck, which is a separate third-party tool. I won't do that for you live now, but I'll just say that once you download those 2 files, in step 1, there's CyberDuck, which you need to go to their website to download, and number 2, the configuration profile. All you do is enter your CyVerse password and you're ready to go. So within about 5 minutes, you should be able to see that working. The way that it looks, just to give you a demo, okay, so the way that that works is you have CyberDuck, which is this tool here you can see in my desktop. I've already connected to the CyVerse data store, so these folders and files are already things that I have on my CyVerse data store. On my desktop here, I have this folder conveniently named FASTQ files. If I open it up, It really contains fake FASTQ files, but hey, okay, this is just an illustration. But you can transfer up to, I would say, 100 gigabytes in about 30 minutes on a good connection, so it really is pretty decent. Once you have those files and you connect it, all you need to do is drag them over into the CyVerse data store. It'll take a moment where it will start to log into that and sort of tell you what it's doing, but then essentially, once it goes in, it's already starting that upload process. These files, these folders are really small, so it's happening quite quickly, but you'd have the option to monitor the progress of it. But really, it's a one-step process, so you should feel pretty comfortable if you have those FASTQ files and they're not in the CyVerse data store. All you're going to want to do is set up the CyberDuck, follow the directions, and move them over. You really shouldn't experience problems, but if you have questions about that or any of the documentation, let's take a look here. I'm still learning how to get all of my windows oriented. Keep in mind, in many places on CyVerse, including our documentation, there's this little help bubble, and you can go ahead and ask a question, and some CyVerse representative will assist you with any problem if you can't understand something or a link is broken. Okay, so let's assume now that you've uploaded some files to the CyVerse data store. Where do you go from there? I have some slides, and we'll think about how many I need to show them. Need to show to you in order to just get you oriented. But you're going to log into DNA Subway, and one thing that you do need to do is make sure that you have a CyVerse account. So you can get a free CyVerse account. It's always free, but it is something that we encourage everyone to do, and that's available at user.CyVerse.org. Okay, so the very first thing you're going to do is create a project. And the steps are here, and I'll give those to you, the slides, I'll post them. So I'll just show this to you, and you can have that for reference. Okay, so let's take a look at doing that. So here is the DNA Subway homepage, which I hope many of you are familiar with. I've already logged in, and to begin a project on DNA Subway, you go ahead and click a green square to get started. Now, there are two options for doing the RNA-Seq experiment. One, either you're working with single read data or paired N. Majority of us, I hope, are going to be using paired N data. That's really good for doing an RNA-Seq experiment, so I'm going to go ahead and select paired reads. Then we have a list of organisms. So one of the requirements for doing the Kallisto software is that you need to have a transcriptome. We have several, I think more than 30 transcriptomes that are available. If you do not see the transcriptome that you want to use, please let us know. We will go ahead and integrate that for you. So please don't think that you're limited to just what you see. In my case, since I'm going to be working with the same data that we've mentioned in the tutorial, I'm going to go ahead and choose human because there are some sample data I know already available for that. So I'll go ahead and click on human. I'll give this a title, and I'll go ahead and click continue. All right. One of the first things you'll see is we've got a little introduction that will already give you links to some helpful pages, including tutorials. So you can read through that, but at the moment, we don't need that. Now, I don't want to make the assumption that everybody on the webinar has used DNA Subway before, so let me just point out one or two quick things. Number one, at the bottom, there is a toggle so that if you need to use the Tuxedo protocol, maybe you're teaching this in class, it hasn't disappeared, it's still there. You can click on Tuxedo and you'll go back to the same implementation that we had previously. So do know that you can toggle. The data underlying are going to be the same, and you have that ability for now. At a certain point, we will no longer support that, but we don't want to take things from folks in the middle of the semester. Okay, the very first step of using Subway with the data that you've uploaded is to go ahead and click on Manage Data. And this is directly connected to the CyVerse data store. So we need to go, and if you had uploaded the data, they would be in your home folder, which usually is your CyVerse username, and you could click on your home folder and browse, and you'd actually be seeing the same folders that we just saw a moment ago in CyberDuck. I'm going to be using the sample data, so I'm going to go ahead and click on sample data here. And I already have sample data for my Zika cells. Also, as another service, if you are doing this in a class and there's some sample data set that you know you constantly want to use, you can share it directly through CyVerse, but you can also let us know and see if we can help you by making sure that that's available for you directly once you go ahead and access any particular transcriptome. So here I have the transcriptome, rather the RNA-Seq reads, and I'm going to select all of these and just click Add Files. It'll take a moment, but then those things will be added into my project, so I'll just give it a moment to go ahead and load that. But like I said, data really shouldn't be a limitation for you. You can use your regular sized RNA-Seq datasets. It just so happens that these datasets are only a few hundred megabytes per FASTQ file. You may have ones that are larger, but in general, the CyVerse data store is quite spacious, so you should be able to upload your large data. Now, it just so happened that the data that we're using, and in many cases I suspect, are paired end data. So we're going to turn this paired mode on, and we just need to indicate to the software which are actually pairs of sequences. This happens to be already in numerical order. So when I pair any two sequences, it will go ahead and make that link just as something that the software needs to keep track of. Okay. So I paired those things. I see the size of the files when they were modified, and now I have a decision point. For the sake of the demo, I'm going to go ahead and start some QC jobs, but I'm actually going to switch over to a project that I made public. I'll show you that in a second, so that we don't wait, because this is only an hour, and in fact, we're only already 30 minutes into it. What you have the choice to do is decide whether you want to run the QC or not. If you know that the files are of high quality, perhaps you've used these in past analyses, you've already done the QC, then you don't have to click this step. You'd be able to continue without it. However, if you want to, or especially if you're teaching, what does it mean to do quality control on your RNA-Seq reads, you probably want to run it. So I'm going to go ahead and click that run link. And what will happen is we're clicking to run a job. You have to do this sequentially, so it probably take me all of 2 minutes or so in order to get these all running, which is why I'm not going to wait for them. But I just wanted to show you that you do go ahead and click run. I'll also mention here that you're limited to a number of concurrent jobs. So the more files that you have, at a certain point, you'll have to wait for some to run before you finish. In my case, running these over the last couple of tries has taken about 15 minutes to get through all of them. Maybe I get through 3/4 of them and I'm over my job limit. I waited 5 minutes and then you can continue. This is mentioned in the documentation, which I'll point out to you, but just have in mind that you can't run every single thing at once. What I'm going to do now is talk about public projects. I'm going to move over to a project which I've already done. In theory, this project is public, so you should be able to see it. I don't know what happens if everybody clicks on it once in the webinar, but it's available for you to take a look at. So that's running. Let me close this and make sure that everybody knows that you have this option here. At the bottom, where I have my project status, right now it says private, which means that only I can see the job. But I could actually change this to public. If I change it to public, it says it's updated. What will happen is that that will appear in this list called public projects. So here it is, public, readily available. People could search for that project by name or by project number, and you get the idea. What we would recommend is that you have a project where you have some data that you want students to take a look at. There's a certain value to clicking links and actually discovering, getting the answer, changing parameters. And when you're doing that, that's fine. But if you just have data that doesn't need to be recomputed, you can easily make your project or ask your students to make a project public, write down the name or just take the name of the student and you can find those. And that's really saves doing computation that doesn't need to be done. I'm going to go ahead and look at my projects. And just another reminder, as you do anything on DNA Subway, it's automatically saved, so you don't have to worry about clicking the save button anyway. I'm going to go back to a project that I've already completed. I'll mention to you that when you have a project that's public, folks can't edit that project. So it wouldn't be that someone go back in and change and adjust that project in a way that makes it no longer or the way that you left it. So let's take a look at that managed data. And when it's actually done, run link will be replaced with a view link. So if I click on view here, it's going to go ahead and load the FastQC report. I don't have a lot of time to go into every detail of what the FastQC report tells you. If there are questions at the end, I certainly can take those questions and come back to this in detail. But essentially, if you look here and if you're familiar with the FastQC report, you can see that the read quality is really, really well. All of these are bases on average are coming up in Phred scores in the upper 30s. So really couldn't get data that is much cleaner than this. What that tells me is essentially I don't have to do any real trimming. If I wanted to, though, I could do that. And so here's the FastX toolkit. I had stopped these jobs, that's why I'm getting an error there. But in the FastX toolkit, you can go ahead and run a job. You can either click run and run it basic, which would be some default trimming parameters. If I click run advanced, I can actually see and adjust those parameters in any way that I'd like. I'm not going to go ahead and do that now. What I want to take you through is what is the next step. So the next step is to go ahead and use the Kallisto quantify. The cool thing about Kallisto is that really there's very few parameters to adjust. So even though, in fact, right now, and in many cases, I'm going to run it without adjusting any parameters, even when you think about maybe I want to change some things, there's not 20 different settings that you need to think about, well, what is the right value for setting A, B, or C, which was the case previously. I'm going to go ahead and click on Submit Another Job. And what I would be presented with when I am doing this analysis are my FASTQ reads. In this case, the left pair and the right pair are appropriately displayed. So the only thing now is to name these samples. So in this case, thank goodness it's saved from yesterday, so I don't have to remember what to call them. But I have these Zika and mock treatments. In this case, I've got 2 replicates of each one, and then I need condition. Now you'll get a warning, but essentially you just want to make sure that you've kept the same spelling and appropriately named your samples. Now, I could go ahead and submit that now, and I will do that. But let me show you the parameters. Again, in many cases, you won't need to touch these, but if you do, let me show you what they do a little bit, show you a little bit about what they do. So the first one that's there is this thing called build pseudo-BAM files. So there is an option with Kallisto, and in fact, there's a new version of Kallisto which makes some of this even better, but there is an option for you to actually go ahead and examine using a genome browser like Integrated Genome Viewer to examine the reads, which are really pseudo-aligned to this transcriptome. That is a sort of, I don't want to say hack, but it's not really a true alignment in the traditional sense. Nevertheless, it may be important or valuable in some contexts to see that alignment happen. So if you want to, you can select pseudo-BAM. We've turned that off by default because actually it's really computationally expensive and the files are really, really big. So by default, it's disabled. If you want to, you can enable it. There are a couple of other things that are here. The fragment length and the standard deviation. These are really only important if you are working with single read data. There's a link here at the top to the Kallisto documentation. If you are using single end data, you'll need to provide these. And these are things that you would know because you set up the experiment or be available from the sequencing company. That's when you'd have to think about providing those. The bootstrap, what happens is that there is a subsampling of your data. You could adjust this parameter. You could adjust it up or down. Adjusting it down makes this go faster. But essentially, you're trying to get an idea or quantify the uncertainty of your abundance. And as you'll see later, when we're presented with some box plots of abundance, where we get an idea of what the error is of our observation, that comes from doing subsampling. If we didn't do subsampling, we'd have no idea. We just have a number without any kind of statistical context to it. You can adjust that bootstrap if you'd like, and the random seed is just there so that you always get the same results for that sampling. I am not at this point going to adjust any of these parameters. There's a couple other things there if you're working with fusion genes or if there's other sort of things, but in many cases they won't apply to most things we're doing. So what I'll go ahead is click Submit. When I click Submit, you notice the way that we've processed things is that those jobs appear as tabs. So I'll give it a second for this to go ahead and process. It'll take a moment. And this step is actually running on the cloud and Jetstream. This could take 20, 30, 40 minutes. It depends on the size of what you're doing. Of course, the number of bootstraps that we've done, 100, which is fairly rigorous, makes it go a little bit longer. but it's still quite quick compared to the other methods. If you look right now, that job 29062, remember that everything that you do in DNA Subway has a job number, and that'll be important to viewing your results later if you want to view them in the context of other places on CyVerse. I'll point you towards that towards the end. Our other job is already finished, so we don't need to worry about it, but just give you an idea that you can have jobs there and you can view the results. So those will also sort of flash and highlight. You can view job info. This job I clicked is already finished. If I go ahead, let's see, and go back and look at a job that's running, if there was some problem with that job, I also have the option at the bottom to stop that job, but I can see the status of what's going on with any particular job. So right now it's just starting. Sometimes it'll tell you that it's running, finishing, but we're a little too early for that. All right, so let's go, and what would you do now that you want to see the results? I'm going to go ahead and click on View Results. And there are a couple of things that you get. So we're in a job that's completed. If I look at this sort of number icon, I can get an idea of the reads that were processed, how many were pseudo-aligned, and what the alignment rate is. So here we have 80%. And interestingly, when you talk to people, that's a really good number. Some people might be expecting or hoping for 100% alignment, but really what you're expecting is somewhere in the 70s or 80s for your alignment rate. It's a factor of pseudo-alignment, but any alignment. It's also a factor of how well the transcriptome is covered in your sample versus the entire transcriptome, right? Certain developmental stages will not be, or disease conditions will not be present for sure. So you wouldn't expect to get 100% alignment rate. If you're getting something that's in the 10s, 20s, 30s, they need to look if there was some sample mix-up. If you notice here, and I'm not going to show it, there is no IGV icon because I haven't done pseudo-ban with you. I'm not going to show that there. It is available. It does take Java, which we're always trying to find ways to kind of avoid because it's a security issue and sometimes doesn't work, but it is possible. And of course, we'd like your feedback on how valuable that is to you and or other things you'd like to see. Now, there is an option here for download. If you click on the download link, you would be able to download a tab-separated file which contains the actual differential abundance testing results. The other thing that's here is this folder icon. And if you click on this folder icon, what's interesting is it'll take you to the CyVerse discovery environment. So the discovery environment, or the DE, is an interface to a lot of things in CyVerse. I'll come back to it in a moment. I unfortunately don't have time to take you through the entire DE, but what I wanted to show you is we're now thinking about it just as a file browser for the moment. One of the capabilities it has is I now have all of the results from that Kallisto job. So if you wanted to use the Kallisto output in a different way, all of those things that you would get if you had done this at the command line are available to you. So we really wanted to make DNA Subway seamless between sort of this interface, which may look a bit simple and sleek and sparse to make it more classroom friendly, but really capturing that this is a rigorous and really authentic implementation of the tool. So you have all of that available to you in the discovery environment. I'll come back to that in a moment to give you some ideas of what you might want to do with that. But for now, I'll return back to DNA Subway. That's what that folder icon does. So what you do next is now you may want to visualize those results in Sleuth or RShiny. So let me go back to my slides for a second. I don't think I have missed out on anything I really wanted to capture. I've showed you public and private projects. We've talked about read quality, and all of these things are available to you in the slides, so I'll post those, and again, you want to remix them for your teaching or for your own edification, go ahead. We're going to go ahead and launch the Shiny app, and the one thing I want to tell you is because the size of the data sets are quite large, it may take several minutes. So I'm going to load it. I have one I think is already loaded if we need to do that, but it'll take a few minutes. What you will see through that is a results menu where you're going to get a tab separated, or rather it's going to be tab delimited, but here you just view it as a really nice interactive and responsive menu of those results, which you can view, filter, or sort. We provide the bootstrap menu, so on a per gene or per transcript basis, you can actually go in and look to see between your two samples what was the level of expression. There's a PCA menu. I'll talk about looking at your sample in terms of principal components. That's important when you get into more sophisticated ideas of thinking about, are you making the right comparison about your samples? I'll talk about that. Volcano plots, loadings, a sample heat map. So we'll get into those. Let me go ahead. Let's go and get that up there. I'm going to go ahead and click, just because I want to see it happen, launch a Shiny app. And as I said, it'll take a few minutes to load. In my experience, it takes somewhere in the order of 3 to 5 minutes. So if you get an error, or if your computer is telling you that this website is slow, you can wait for it, as it's doing now. We will be working on a way to optimize that, but still, all things considered and the amount of data, it doesn't take all that long. Let me go ahead and load one. I'm going to have to reconnect to it. But theoretically, and I notice it's a bit browser specific, because on the Safari browser, it seemed to work really, really quickly. So give it a second. But the idea here is a couple-fold. So let me explain that while that is loading. This is a technology using the R language called R-Shiny. So if you've ever seen some sort of interactive infographic, perhaps on a website where you can make adjustments, this is what that application gives you the ability to do. And the good thing about it is that if you are using something a little bit more sophisticated with your students, maybe you're teaching them R, you want to think about data science, that you can actually go ahead and use these results and explore them in R yourself. The difficulty of using R is if you don't know how to use R, you're not very comfortable with it, it can be a lot to get started. And so generating visualizations and graphs of things that are going on may actually be a little bit difficult for you to do. And rather than making you learn those command line things in order to generate them, what this gives you is the option to sort of have them in a point-and-click way. I'm going back here, because I'm just going to make sure that they're loading, and we'll see which one loads first. This one only took a few seconds before, but I know it's trying to reconnect. And in fact, I hope, because I'm connecting to two implementations at the same time, as I said, it's going to take 2 or 3 minutes for it to load. I hope that I'm not loading and making it slower, but we'll come back to it in a moment. What you can also do from here is you can also do your filtering and downloads straight from the R-Shiny app as well. So just give it another moment with me. I'm waiting for this one. And while I'm waiting for that, I'll go back to look at questions that we've gotten so far, because I know we had one at least. And if there are more, I'll take those while we're waiting for those to go. So let me just go back to the chat. And if you have questions now, it's also nice go ahead and do that. This one is more of a statement than a question from Doug about prostate cancer RNA-Seq and loads of data on different sized tumor and normal tissues. So I'm assuming you're maybe asking questions, Doug, about whether you can use this yourself. And really, yes, you can. You may want to think about how you separate those experiments into something that makes sense. You can actually do multivariate comparisons using Kallisto and Sleuth. So let's say you have a tumor sample and you want to look at a genetic background. Maybe you have several genetic backgrounds of that tumor. Or let's say that you have that plus you have a treatment of a drug and not a drug. You can do that. Purposefully, because that gets a little bit complicated, we decided not to make that possible directly through DNA Subway. So what would be smart is actually to go ahead and use Subway for a simple condition, and then graduate users to using Kallisto and Sleuth, perhaps in the discovery environment, which I'll show you in a minute, or in a Jupyter or RStudio notebook. I'm going to show you those in a moment. I'll take 2 more questions, and since my thing is loaded, I'm going to return back to that. One of the questions was, will I post the slideshow? Absolutely, and you'll get a link. I'll show you that. And also the next question was organisms that are polyploid. Yes, you'd be able to work with polyploid organisms. The key requirement is that you have a reference transcriptome available. And if you have more questions on that, I can help you towards the end. But let me take 5 minutes to walk through the Sleuth results, which have loaded, and then we'll talk about any other questions that we get to. But please go ahead and either put those into the chat or the Q&A, and we'll come back to those questions. Okay, So this is loaded. I don't know if they both loaded. As I said, it seemed to go faster in Safari than the other one, which is good for me to pay attention to. But here are my results. And I can go ahead and page by page go through any results. I can go ahead and sort. If I want to have beta, which is a corrected p-value, and sort of search in that way, I can go ahead and search that way. And I just wanted to pay attention, sorry, to the chat. I want to make sure I'm in full screen here so I don't have access to everything. Okay. You can go ahead and sort them in that way. On the left-hand side are some explanations of things. Like if I want to see the variance of something, I can add that as a column, and it'll go ahead and pull that up. If I want to search for a gene, Doug was mentioning prostate cancer. I used to work on that. So if I search for P10 and I want to have an idea of what was the differential expression there, I could go ahead and do that, or look at something more you know, basic like tubulin or whatever the case is. You can go through and sort and filter on your genes in a dynamic way. So it makes it really easy. And keep in mind, it says that there's 55,000 rows. This would actually start to crash on Excel. So it works pretty facile here. The next menu, and these are all explained, although I'm going to go through them at a fairly fast clip so I can get to questions. Keep in mind, these are explained very well in the documentation. If I go to Bootstrap, I can, on a per gene or per transcript level, look. So here is my mock condition in red. Here is my Zika condition. If I click on these, I can also save these images. So you could have assignments where your students are asked to look at certain genes, and they could go through. And then on those basis, they could search for whatever type of gene. If I go through and I want to, type in a gene, I might need to actually put that there, show genes, and then type in something. I could also do some kind of searches too, but I have to go back and copy and paste because it wants the transcript name, but you get the idea. The other thing is a principal component analysis. This is really important quality control. So although I can't explain PCA in 1 minute, one of the first things you need to think about when you're doing an RNA-Seq experiment is, are your replicates more similar to each other than they are to your treatments, or the difference in your experiment. So what we're seeing, although there's difference, the mock are on one side, the Zika are on another side. We could use some more replicates here to bring these closer, but you might want to study this in order to think about what are some confounding variables and variation that may be brought in your RNA-Seq experiment. For a volcano plot, again, you could do some searching. If you don't enter Pacific transcript, it would give you the ability on the plot where we can zoom in or out or pan to see overall truly differentially expressed according to the test here versus not. And you can go in and zoom and pan and play with those. On the loadings, you can actually see for any given principal component what were the individual transcripts, which are the most variable in your sample. And you could copy and paste those and go ahead and search for them in your results. And you also have a heat map, which in this case is giving you another pairing, an examination of your samples and your replicates. And this is a quality control step that gives you that option. But really with those results, which may be the main thing, you can take these, examine them, download them, play with them, and explore them elsewhere. I've got about 10 minutes, and I want to get to as many questions as I can. So let me show you just the last few things I want to show you, give you some hints, and then just use the rest of the time for questions. So where to go from here? So one other thing to look at is this really nice project we put together, and many of the folks attending perhaps today participated, is this RNA-Seq for the Next Generation course. There are some sample data sets that are available there, but really was a project done mostly of faculty at primarily undergraduate institutions. So these faculty are I would say, sort of say, pioneers of folks who have combined research and education by using RNA-Seq in the classroom. They used and helped us test and build the original Green Line. We'll be relying upon their feedback for improvements now. And in many cases, maybe you know some of them, or they'll be happy to hear your feedback or contact for advice. Definitely pay attention to the survey we're going to send out after this webinar, because we want to get your feedback on actual making improvements to the Green Line, making it useful to you. The resources on learning that CyVerse I'm going to point out to you in a minute. I'm going to talk about this thing called Vice in 160 seconds. So if you are used to or thought about using Jupyter Notebooks or RShiny or RStudio, I'll show you how I can use that to extend what we've done into a real data science. So extend that from Subway. And one sort of teaser is that we hope to have another webinar in the not too distant future on the Purple Line, which you may have seen, especially if you used DNA Subway for a while. It's the latest thing. So we're going to be using a Purple Line, which is a classroom-friendly implementation of Chime. Very similar in many ways that we've made those optimizations to the Green Line. We've done many of the same things for Purple Line and for Chime. That's for metabarcoding and eDNA. So if you're looking at microbial diversity, microbial populations, or even looking, let's say, in a water sample where you want to look at vertebrate diversity using environmental DNA. We also, as we've tried to do with Greenline, pair that with wet lab protocols, which reduce the cost and make it really efficient for the classroom. So stay tuned. Let us know if you're excited about that. We'd love to point you in the direction of more resources. One last commercial before I show you those last two things and then take your questions. If you are faculty interested in getting some more computational skills, pay attention to the CyVerse website. We have 2 extended training programs. The Container Camp, so if you're working with reproducible workflows, think about that happening quite soon, March 6th through 8th. But also, this new course, it's going to be 2 weeks on foundational open science skills, and it really will be even more than a boot camp. We really want to go in depth with faculty members, training them on the computational skills that they need to do effective, high-impact science and education. Look at the CyVerse website for more information. We're taking applications quite shortly. So acknowledge CyVerse, and quickly let me show you one last thing, 2 last things, and then go straight to questions. So the 2 last things on learning.cyverse.org. Let's go here. On learning.cyverse.org, if you go to the DNA Subway guide, there's a manual for everything in DNA Subway, including the alpha version manual for Purple Line if you're really super excited. But if you go on the Green Line, there is a walkthrough in detail of everything I have presented. So you can really take that and use that at your own pace, circulate that. There's also a brand new 5-minute video that walks you through DNA Subway Green Line, just the highlights. It's less than 5 minutes. It's a quick recap to orient you through everything that you've seen there. So take advantage of those learning materials. I mentioned on the discovery environment, there's this new application called Vice, just to give you in 60 seconds what you can do. I started a Jupyter Notebook. Let's go ahead and load that. Let's call it webinar. And see if that will load. Oh, I've got it open up twice. I don't know if this will fix it, but I've got the other one open and one of them will work. If I don't go that one, I'll go this one. What I've done, oh, I did have it loaded. Never mind, I don't even need it. Oh, that's why, okay. I've loaded some notebooks that I use in teaching researchers how to do Kallisto RNA-Seq. And so if you're familiar with the idea of Jupyter, you can go ahead and create Jupyter notebooks which go through, your data sets. And you can bring in data that's your own data or data that you've partially pre-computed for Subway, so you can mix and match. But you do have the ability within CyVerse to go ahead and launch things in Jupyter. There's a webinar that's been posted to YouTube, so take advantage of that. It really takes things in a more sophisticated direction if you're now moving from what we hope are freshman and sophomore experiences, course-based research, where once people get the familiar, the fundamentals using the Subway, then you can take them and do even more custom things. So I showed you a lot, and now I'm going to get to as many questions as I can. I'll even go beyond the hour, but I don't want to have any main content out of that hour. So I'm going to stop sharing. Well, let me go ahead and look at some of the questions first. I'll look at the chat first, and then I'll go back and take a look at anything that you need to see. So somebody mentioned, does the reference transcriptome need to be annotated? Does not need to be annotated. It's to your benefit if you have one. May make some of the analysis easier, but it doesn't have to be an annotated reference transcriptome. The next question is, are the dates wrong? And you're correct. If it said June through 7th, that's a typo. It is starting that first week in June, so maybe that's supposed to be a 17. It ends earlier than that, but it's about 2 weeks, so thank you for that. Let's look at these other questions. Okay, so are publication quality plots or graphs downloadable from RShiny? Yes. Any of those plots, you can right-click and you should be able to download them. If you have any problems, let us know. For small RNAs is the next question I have. I would have to, so when as long as your transcriptome contains small RNAs, that's good. And I don't remember if there's any reason why you shouldn't be using Kallisto with small RNAs. You may need to think about how you adjust Kmer values. So I would Google Kallisto small RNAs. It will do anything that Kallisto does. I haven't used it for small RNAs. There may be, as you already wise to ask, some caveats for that. Next question I see, is it possible to compare multiple conditions against a control, let's say normal cells versus different for 4 or 5 mutant cell lines against everything? So this, I think I might have answered before. It is possible in Kallisto to do multivariate conditions comparisons. We have specifically not enabled that in DNA Subway. We've made it so that you really are doing an A-B comparison by our choice. So that's a design decision that we made. However, if you're using Kallisto in general, you can go ahead and do multivariate comparisons. It's more difficult to set that up in Subway, but it's not super difficult to do it in Kallisto. You have to prepare an experiment design file, which is just a text file where you have all your conditions, but it's really all done in R. I have notebooks and other folks have notebooks that demonstrate how that works. Does this work for bacteria? If you have a bacterial RNA-Seq experiment, in principle, you'd be able to provide a bacterial transcriptome and your files. We're not worrying about splicing here so much. The data sets are already available in DNA Subway. So that's the next question. Where can I find a data set to upload? You can we provide some sample data sets, as you saw with the human example. The other place to go for sample data, if you need some for teaching, on that RNA-Seq for the next generation website, there are some data sets there. And also there's a tutorial on learning.CyVerse.org about importing data from the sequence read archive. I might be able to bring that up quickly. So on the CyVerse Learning Center, there are some tutorials, import data from NCBI SRA. So you could follow this tutorial and it would show you how to bring data in from the SRA. So you could go into the SRA, which I promise is not the easiest place to search for sometimes, but you can do it and search for some RNA-Seq data sets and use those. Okay, answering as many questions as I can. Next question. In the classroom, I'd like to have students do the same analysis so I can run the analysis before and know what to expect. No, that's perfect. Does this go against the use of resources? That's exactly what we'd like you to do. Go ahead and analyze it yourself. Make that project public. Then use your own judgment as to what you need your students to do and what is just reanalysis. Our main message is that we just don't want folks to think that they should use it with 70 people all analyzing something at the same time, and it's been done every single year. Try to use some wise use of the resources. We'll be monitoring and see how much we can provide, but we really appreciate it when our users pay attention to that. Can you upload your own de novo assembled transcriptome? No. However, you can provide it to us and we can work with you to put a transcriptome up there that's not provided. I'll also remind you on the CyVerse data, the Learning Center, there is also a tutorial on using Kallisto, and let me go to the home. There's a tutorial on using this in the discovery environment, which is a lot more flexible, and you can go ahead and use that on your own anytime and upload whatever you'd like to work with your transcripts. The next one is, can Kallisto map Kmer's multiple splice isoforms or quantitation? Yes. If you have multiple isoforms, it will go ahead and automatically take care of those. Repeat question, for reference genomes you don't see, although we don't allow you to upload that yourself, we, at the moment, are doing it manually. So you can use, let me make sure everybody knows, on DNASubway.org. At the bottom right-hand side, there's that contact us link, and just let us know about what you need and give us that feedback, and we'll go ahead and do that for you. If a reference transcriptome is not available, I think that that's a repeat question. So I'm going to stop sharing for now, and I'll just take another 30 seconds if anybody has questions that they haven't provided for us in the time that we've had allotted. I'm just giving people another chance, last chance to pull up any questions they may have for us. One thing that I'll remind you is that we are anxious to see people use this and really get feedback from them on what did they find most useful, what do they think that we could do to improve that. Any type of feedback is really, really appreciated. You will be getting a survey link from us, and I'm going to go ahead in the chat and also put that survey link there for you. So if you look at the chat as you're going, that's the survey that we're going to also send by email, but please go ahead and give us 5 minutes of your time. That's why we do this. And